All right, who's ready for a neuro review? Let's see. So, move my noisy chair slightly. Okay, let's get started. Oh, one thing I need to do, which is turn on this laser pointer. Okay, so the first thing we're going to talk about is cerebral blood flow. Usually, this is one of the most confusing subjects, so that's why we're going to start here, so you can, come back to it a couple of times if you need to. So where do the ACA and the MCA begin? Actually, you know what? Before we answer these questions, let's just take a look at this circle of Willis. This is actually, I think, the spider of Willis drawing that I have here, because it looks a little bit more like a angry spider. It's the same thing, right? The circle of Willis is the spider of Willis. It's just one is slightly more anthropomorphized, and the other one is just blood tubes. Okay, so we've got blood flowing in mostly here from these two vertebral arteries and this anterior spinal artery. They're all going to come together. They're going to anastomose around here. And so right before they come together, you've got some cerebellar arteries coming off the posterior cerebellar artery. You're not really on the hook for knowing where the posterior cerebellar artery comes from. Just know that it's another thing that can be served from the same general area. Then after they come together, you've got the anterior inferior cerebellar artery coming off of there, and everything has now come together to form the basilar artery. So if you actually look at the other pictures that I have of these in the lecture, you see that when these three arteries come together, the left and right vertebral arteries and the anterior spinal artery, when they come back together, it makes a pretty large basilar artery. So there's a lot of blood flow in that area. So we have the basilar artery, it comes up. The superior cerebellar artery is going to branch off of that as well. And then at the very end, where the basilar artery is just going to kind of dead end, like a road. There's a road up here by my house that is a pretty large road. It's Power Road, if you know of it. Although most of you are online, probably not in Arizona. And even if you are in Arizona, you probably don't know Power Road. Whatever, everybody's got these roads near their house, right? So it comes up. It's a pretty large road coming down, and then it just dead ends. You can't go further than that. It turns into state land or possibly federal land after that. And so you have to turn one of two ways. Well, because it's such a large road, people come down and then they'll branch off. It's a large road, and then it just kind of like turns into nothing, but that's because you branch off into one of these two directions, and that is a large road. That's kind of what happens with the basilar artery here. So the superior cerebellar artery is coming off of that. It's superior, so it's near the top, but it's the cerebellum. Remember, the cerebellum is below the cerebral hemispheres. So what the basilar artery branches off mostly into, these large parts, is the posterior cerebral artery, left and right, posterior cerebral arteries, which is what we have right here. And then you can see that it kind of stops, right? We've got these little hash marks here. This is for one of the posterior communicating arteries. My pipes in my house are knocking. It's always jarring. And it might not have even come over the recording, so you probably just thought that I freaked out for a second. Anyway, if it did, I need to figure something out with those pipes. Anyway, you have the posterior communicating artery coming up from the end of the basilar artery or the beginning of the PCA. And that's going to come up to the carotid artery right here. We've got one on each side, right and left carotid arteries. The carotid arteries are coming from, other parts of the body. And by the time they get up here to the brainstem, they're going to kind of dead end as well. And they're going to turn into, let's just talk about the right one. Yeah, let's talk about the right one where I am here. So we'll talk about the right carotid artery comes up. It's going to dead end as well. There's not a carotid artery that continues from here. It turns into the ACA, the anterior cerebral artery, and the MCA, the middle cerebral artery. So it's going to come up and it's going to branch as well. So the basilar artery, there's only one of, right? It's not a right and left kind of thing. It branches into a right and left PCA. We do have a right and left carotid artery coming up, and it's going to branch into either one. So the right carotid artery is going to branch into a right ACA and a right MCA, and the left carotid artery is going to branch into a left ACA and a left MCA. Okay, it's kind of a tongue twister when you're talking about it. You've got the anterior communicating artery here and the posterior communicating artery here. This right here is the circle of Willis. The spider's head is the circle of Willis, right? This is the redundancy, which means that if there's a blockage anywhere, say there's a blockage here, say the carotid artery is blocked off before where we're seeing it here, so like a little bit further down in the carotid artery. If there was no communicating artery, there would be no blood coming to the MCA and the ACA. So the right carotid artery is blocked off. No blood flow gets to the MCA and the ACA. Except that's not true, because we have these communicating arteries. So some blood from the PCA is going to be diverted up here, and also some blood from the left ACA is going to be diverted over here through this anterior communicating artery and through this posterior communicating artery. So you have this kind of redundancy effect. Similarly, if there's a blockage here, that's okay, because blood is diverted from the left ACA, and then it comes across the anterior communicating artery, and then it serves both of those hemispheres as well. It's important because the brain is it's what drives everything. You can't live without a brain. So you have to have blood flow coming to the brain. It makes sense that there are these redundancies in these areas, and not in, say, like the fingers or something like that. All right. So where do the ACA and the MCA begin? Well, you should be able to answer that now. We've talked about it. They begin at the carotid artery. What is the pathway for the MCA? We didn't talk about this, but the pathway for the MCA is that it's going to come off of the carotid artery, and then it's going to move between, so on the superior surface of the temporal lobe and the inferior surface of the parietal lobe, it's going to come out between those, kind of like if you imagine this as the temporal lobe and this as the parietal lobe, then you get the MCA coming out between those, and it'll branch up and it'll branch down, and it'll serve the temporal lobe and the parietal lobe in those areas. Other parts as well, but that's the main pathway that you need to know for that question. Where does the PCA begin? Basilar artery, right? If you already have taken 311 or you're taking it concurrently, don't get the basilar artery confused with the basilar membrane. Two very different things. Okay, so what branches from the basilar artery? The PCA, obviously, but since we've already answered that question about the PCA, what I'm looking for here is also the cerebellar arteries. You don't need to know which ones for the exam, but know that the cerebellar arteries are also branching from the basilar artery. What function do the communicating arteries serve? We answered that as well. They help create that redundancy of blood, so that there's never a loss of blood flow. Now, of course, what if you have a blockage here, like beyond the circle of Willis? Then those communicating arteries and the redundancies won't help. But, it can only do so much. It's not like you're going to have redundancies all throughout the brain. That would take up far too much space. The brain would be mostly arteries and not brain. We need that space for brain. So, it's not going to, of course, we're going to still have strokes. Of course, we're going to still have problems like that, right? But ischemic strokes is what that would be when you have a blockage, not a hemorrhagic stroke. So you'll still have ischemic strokes where there's a blockage in distal parts of the ACA, MCA, and PCA, but you will kind of avoid some of these large-scale problems, like entire hemispheres of the brain going out because of a blockage, because of those communicating arteries. What provides the blood-brain barrier? Well, I hope that you remember that this is kind of a two-part question. There's two things that provide the blood-brain barrier. One is the ependymal cells of the choroid plexus. Blood comes to them, and it creates cerebrospinal fluid from the blood. That's blood-brain barrier. The other one is astrocytes. All right, so astrocytes, ependymal cells. These maintain the blood-brain barrier. All right, next we will talk about the meninges. All the different layers that you need to know, they are on this picture, and so let's just go through these, right? So this layer right here, what do you think this layer is? Notice that it comes down here, it kind of goes around like this. What do you think this layer is? If you said the skull, you're right. And now we'll actually talk about meningeal layers. So we've got this one here under the skull. Do you remember which one is under the skull? It's the dura mater. Under that, you've got arachnoid mater, which also means spider mother, which is like that should be the name of a metal band for sure. Spider mother, doesn't that sound like so cool? Anyway, underneath that is the pia mater, and that one is very tightly adhered to the actual surface of the brain. When you look at this, you really can't tell, is there pia mater or is it just brain? There's pia mater here. Pia mater is always right on top of that gray matter of the brain. So we've got pia mater, arachnoid mater, dura mater. All right. What about in the spinal cord? Are the layers different? Yes or no? No, they're not any different. There's a slight difference with the dura mater, but it doesn't change the order of layers at all. And in fact, how is the dura different in the skull and the spinal cord? You remember? In the spinal cord, the dura isn't attached to bone. It's just a dural sac. Sorry about that noise. Had a notification. Okay. So it's just a dural sac in the spinal cord. It's not attached to bone. But the order is exactly the same. How does the dura relate to the cerebral hemispheres? This, I think, is kind of like a weirdly worded review question. We're just going to jump right into it. The dura mater helps separate the cerebral hemispheres, right? So you've got all of those different sort of plates of dura mater that'll come down. Falx cerebri separates the cerebral hemispheres. It also helps elevate the cerebral hemispheres above the cerebellum. So it just helps create those divisions. That's how the dura mater relates to the cerebral hemispheres. The point of attachment for the spine, this is not a meningeal layer, is what I'm talking about here. I'm actually talking about bone here. So like, what is the point of attachment for the spine? How does it get into the skull? Do you remember what that's called? It's called the foramen magnum. Literally means large hole. They're very creative. In fact, when we get further into what we're talking about in this anatomy class, you'll see that they just use foramen everywhere. Like there are mental foramina, which not like mental, but mental is in like the mentis muscle or mentis process down here in the mandible on your jaw. There's two little holes that allow nerves to come through, and they're called mental foramina. And there's all sorts of other foramen. And so they're like, just call it a hole. It's fine. I mean, it is a hole, right? It's fine. It's just it seems somewhat, almost too laid back. They're like, I don't know, it's a hole. It's a large hole. Call it large hole. How is the pia different from the dura and the arachnoid mater? We already talked about that. It's very tightly adhered to all the different layers of the brain. So where the arachnoid mater and the dura mater are kind of like a grocery bag, like you just threw a brain in a grocery bag, the pia mater is more like one of those food saver bags. When the vacuum sealed ones, and it just kind of like, and you have a broccoli in there, but the plastic is like just over the broccoli, and it looks like it's a fake plastic broccoli because it's just so tightly adhered to it. That's what the pia mater is like. The dura mater and the arachnoid mater are like grocery bags. Okay, where does cerebrospinal fluid exist? We have like 2 videos on this because I like to be redundant about cerebrospinal fluid, and so probably you know the answers to all these questions. Cerebrospinal fluid exists in the ventricles, right? And in the subarachnoid space. What organ produces it? Which one? The choroid plexus, of course. And what cells make that up? Ependymal cells. Ba-dum-bump. We've already talked about that in this review. Covered it like maybe 3 times in the class. I'm sure you got it. I'm sure you got it without me going back over it. But, it's okay. We'll do it again anyway. What is the path of cerebrospinal fluid? Well, we've got a whole video on that. But in general, the path of the cerebrospinal fluid is most of the cerebrospinal fluid is going to be produced in these lateral ventricles right here. From the lateral ventricles, it's going to travel down to the donut-shaped third ventricle. From there, it's going to come down the cerebral aqueduct to the fourth ventricle. And here, from the fourth ventricle, it's got 2 choices. It can follow the spinal cord and kind of bathe the spinal cord in cerebrospinal fluid, or it can go out into the subarachnoid space. When it goes out into the subarachnoid space, it's going to pass through the arachnoid granules and be reintroduced into the bloodstream. And that's it. It's continually pulsing around. That's one other thing the cerebrospinal fluid is. So the cerebrospinal fluid is always moving. Cerebrospinal fluid that does go down into the spinal column will eventually come back up. The cerebrospinal fluid is replaced quite often. You're going to have to check my lecture about this. 7 hours is, I think, when the cerebrospinal fluid is completely replaced in the brain. One other thing. Let's jump back really quickly to right here. What provides blood-brain barrier? There's one other thing, actually, that provides the blood-brain barrier. So we have the actual barrier where blood comes up to an area like the ependymal cells and it's filtered into cerebrospinal fluid. We have another area where blood comes up to astrocytes and then nutrients from the blood are passed through astrocytes to the neurons. The other area that helps keep the blood-brain barrier intact are the granules, the arachnoid granules in the subarachnoid space. They're kind of like a valve, right, that will only allow travel one way. Cerebrospinal fluid can come out, blood can't go back in. And that's due to a pressure differential. Blood won't be able to travel back into the subarachnoid space, but cerebrospinal fluid can come out. So really you have like kind of a 3-part system there. Okay, what does CSF do? Well, CSF does quite a few things. I mean, one thing is it carries nutrients to the brain. It removes waste products from the brain. So that's a pretty important thing. Then the other thing that it does, this is the second part, is it helps keep the brain afloat. The brain would be so heavy that it would kind of smother itself under its own weight without cerebrospinal fluid. So cerebrospinal fluid helps kind of float the brain. And then lastly, it also helps cushion the brain from different kinds of blows. Obviously, we get concussions and stuff still if you're boxing, if you're playing football, if you also just fall down really hard, stuff like that. You're still going to get a concussion, but the cerebral spinal fluid does help cushion more tame kind of injuries like that. Okay, so now we're going to move on. We're just going to name some areas in the brain. I'm going to point to them with a giant green arrow, and you're going to say what they are. Ready? All right. What is this? That's the internal capsule. One way that you know that this is something that is like a communicating part of the brain is that you can see how much more pale it is than like cortices is what I want to say, right? So we can tell that these are white matter pathways because they're more white. They're more pale. If you look here at this one, which I think is my next question, what is this? This is like the main white matter pathway between two hemispheres. Any ideas? The corpus callosum. So the corpus callosum, you can see just how pale that is in comparison to, say, like, look at this. This is a lot darker. Over here at the ends of all these, where we actually have the gray matter areas, this is a lot darker. Of course, it's not gray and this isn't white. It's more like these are all various shades of tan. Various shades of tannish pink. But you can see that it's more light colored. And that means that it's a large communicating area. There's a lot of axons flowing through there. There's a lot of myelination, dense bundles of neurons. And so these are communicating areas. Well, the communicating area that heads down towards the spinal column is, of course, the internal capsule. So that's that one. And then the one that goes between hemispheres is the corpus callosum. Okay, what do we got next? What is this? Boom, boom, boom. So what we're looking at here, what I want to know is it is a lobe. It's a part of the brain, a major part of the brain. So not the frontal lobe, not the parietal lobe. Which one? It's the insula, right? Because that one's kind of hidden underneath the other parts of the brain. It's hidden beneath the parietal lobe and the temporal lobe. It's back in here behind these. If you were to stretch them open, you could see it in there. And the reason why it gets its designation as another lobe is because it looks like a lobe. It has the wrinkles that you would say, oh yeah, that's a characteristic of different lobes, very important computational parts of the brain. So we've got that up here for the, well, depending on where this slice is, it could be frontal, it could be parietal. So whatever we're looking at here, you see those wrinkles here. It's definitely temporal, so you see those wrinkles here, and you see those wrinkles in the insula as well. So that's a characteristic hallmark of what makes up a lobe. Next, this. Nope. Oh, this. This is a ventricle. It's just, it appears as a hole in the brain when we've taken this coronal slice. So, that was a question that I was going to ask you. Man, shoot. What kind of slice is this? It's a coronal slice. What about this one over here? What kind of slice is this? Think about that. We'll talk about it in a second. So the ventricles in a coronal slice just kind of appear like empty holes. Of course, any cerebral spinal fluid that's in there would be drained out. And so it really does just kind of appear as a hole. But that's one of the ventricles. Okay, so this is a sagittal slice over here. And I think we're going to move to that on our next. Whoops, nope, I'm wrong. Okay, so what structure is this? This might be a little harder. It's a dense bundle of gray matter in the bottom portion of the brain, more medial than the internal capsule. It's the thalamus. That one's a little harder to identify, but that's the thalamus there. Okay, so now we're going to move to the sagittal slice. What do you see here? It's the corpus callosum again. This is just the anterior portion of the corpus callosum that I'm pointing to, but you can see how much more pale and obvious the corpus callosum is than anything else. That's because, again, of those dense bundles of neurons, of neural axons that are traveling through that area. Okay, next, right here. This is the PCA. I mean, you can tell how large that is because, look at this. We're not under magnification here at all, right? This is just someone holding a brain. This is looking at a brain that's about the size that it is from someone's head, and you can easily see the PCA here, which means this artery is huge. And largeness in terms of an artery, pretty much goes right along with importance. So it's a large, important artery. This is the PCA. It's towards the back, providing blood flow to this posterior portion of the brain. And what I'm looking at here is this over here. This looks like another artery. Of course, it's in the cerebellum, so it's one of the cerebellar arteries. I can't tell where it comes from, so I'm not sure which one it is. You're, of course, again, not on the hook to know any of the cerebellar arteries, just that they exist and that they come off of the basilar artery. But that looks like one of them to me right there. Really quickly, again, you can also see that down here in the cerebellum, you have this branching structure again, and you've got these white matter axons that you really, really, really can see pretty clearly. Okay, next. This, I'm talking about the covering over here. To me, this looks like arachnoid mater. Why does it look like arachnoid mater? Because it is not tightly adhered to the surfaces of the brain. I would say that this is all pia mater over here. This is arachnoid mater here because it's more like, again, that shopping bag kind of look. What is this? I've totally given this one away. It's the cerebellum. What is this? This is a part of the brainstem. It's a large, kind of knobby part of the brainstem. It's the pons. What is this? This is another artery. And you're probably thinking, well, is it ACA or is it MCA? I'm not sure. I think that it's MCA, and I will tell you why. And it's kind of hard to tell because it's cut off, so we can't totally see where it's going. The ACA should come right up along the front of the corpus callosum. That's what should provide blood flow to the corpus callosum right here. So it just kind of comes up along this front portion, rides right along it. Now I think what we're seeing here is this is like the left MCA, and this right here is probably the right MCA that is still hooked on and going to go. Well, that actually doesn't look like an artery to me. Let's forget about that. But this guy here, my guess is that it's going to come towards us. It looks like it's facing that direction, and it would then go between the parietal and temporal lobes. You're not going to have guessing games like this on the exam, of course. You're going to have obvious ones like the corpus callosum, like this PCA back here, like the cerebellum. So that's just kind of to orient you with these arteries and show you how absolutely large and important they are. Okay, so that's that one. What is this? This is an important question. I'm not talking about the part of the brain. I'm talking specifically about this kind of like large hill of brain goop. What is that? It's a gyrus. And what does that mean that this is? A sulcus. Exactly. So we've got gyrus, sulcus. Gyrus is a valley. Gyrus is a hill. Sulcus is a valley. And what is this? It's that person's hand. Definitely not going to be on the exam. Okay. So let's talk about the anatomy here really quickly. What is Broca's function? And that's right here where we are. This is Broca's area. Broca's function, I mean, let's just talk about it in really brief, easy terms, speech production, right? What about Wernicke's function? Where's Wernicke's area? It's not the next one. It should have been. Wernicke's area is right here. Wernicke's area is speech comprehension. Of course, like I said, more recent articles are showing that there's a lot of different areas that are involved in speech comprehension, that Wernicke's area might not be something that in the future we even talk about. However, it'll take a long time for that to end up in practice, in the textbooks and stuff like that. So for now, you should know Wernicke's area is comprehension. But when you see it change someday because of more studies and that idea becoming more known, more widely known, and put into textbooks, don't be surprised. So for now, Wernicke's area, comprehension. What connects the two? The Broca's area and the Wernicke's area. Do you remember? It's a connection pathway called the arcuate fasciculus. The arcuate fasciculus is what connects the two, and you should definitely know that. What happens with damage? Well, damage to the arcuate fasciculus is going to impair mimicry. Damage to Wernicke's area is going to impair speech comprehension. Damage to Broca's area is going to impair speech production. And what is functional mapping? Functional mapping just means the brain is mapped out in accordance with function. So examples of that are here in the primary motor cortex and the primary sensory cortex. Remember that if you look up here, this is like about the knee or foot. Now foot is down in the middle. So this is like torso, possibly upper leg type areas right here. We've got arms, and then we've got face down here for somatosensory. This is motor. It's laid out exactly the same way. And interestingly, the part of the motor cortex that corresponds with the movements of the face and the tongue and all that good stuff is part of Broca's area. Speech production. Makes sense, right? Other parts that we can talk about functional mapping is the auditory cortex. Remember that it's laid out low pitches over here, low tones, low frequencies, and then high tones are going to be over here. I don't know if it's strictly like that. I think it's a little bit turned, but in general, you've got this like tonotopical mapping. And then back here in the visual cortex, you've got this would be the upper part of your vision of what you're seeing out in the world. This corresponds to the top of the world. This corresponds to the middle, and this corresponds to the bottom. And the closer you get, the closer it is to the center. So that's functional mapping. Okay, so let's do some naming of areas. What's this? Oh, we already talked about that. It's the motor cortex, right? That's important for motion. You know, it's going to initiate all the motions, speaking motions, walking motions, sign language motions, all sorts of, any motion is initiated in the primary motor cortex. It is right next to the primary somatosensory cortex, and that makes sense because you're going to have motion and you're going to have feeling, and a lot of times you're moving and you're touching something. So like if I'm touching the top of my microphone right here, there's motion, and then I'm touching something. You want to put those together because you need to know how to control the motion. So if you're feeling pain, you're not going to do that motion. If you are not quite on target with what you're grasping, you're going to need to, change and get more on target. So that's why those two are next to each other. The other thing to know is that the frontal cortex stops right at this division, right? So the frontal cortex is all this here in the pinky, purpley colors, and it stops right here between the motor cortex and the sensory cortex. Okay, this is the primary auditory cortex, also known as A1, also known as Heschel's gyrus. You should know all three names. Primary auditory cortex, A1, which just means auditory 1, primary auditory. It's really the same name. And then Heschel's gyrus is the other name for that. There's the arrow that corresponds to Wernicke's area that should have come earlier. There's the arrow to the primary somatosensory cortex or sensory cortex. Either one is fine. And that's it. That's all we're identifying in the brain. On this picture, you should know that this is the occipital cortex back here, and this is the temporal lobe, temporal cortex here. Of course, the insula is buried in about right here, and you can't see it in this picture. Okay, next, we're going to talk about central nervous system and peripheral nervous system. Which one is encased in bone? This is one of the first things that we learned about in this unit module thing. The one that's encased in bone is the central nervous system. It's inside bone. It's in the skull. It's inside the spinal column. The central nervous system is in bone. The peripheral nervous system is outside of bone. What cells are in the PNS? We're just going to move to the diagram over here because this is going to answer that question for us. So what do we got here? We're going to go through a neuron. A neuron, of course, is in either the central nervous system or the peripheral nervous system. But let's go through. We'll take a slight detour and talk about parts of the neuron, and then we'll be back onto what kind of cells are in the PNS and CNS. So what part of a neuron is this? Dendrites. That's right. These are dendrites. What about this? It's the body of the cell, which is also called the soma. So this is the soma of the cell. This is, of course, the nucleus. This is kind of like, I think, high school biology, so you probably know most of this so far. This is the axon. Good. And then down here, these are the teleodendra, right? So these are kind of, sort of like dendritic spines, but they're the end parts. They're after the axon. They're also called teleodendra. The very end of them is a terminal button. This is where neurotransmitters are released. And the area between the terminal button and the dendrite of the next neuron. Wow, that was... totally lost the word neuron. That was weird. So the area between the terminal button and the dendrite of the next neuron is the synapse. Okay, so that's a neuron. It exists in both the CNS and the PNS. There's something on here that we don't have a picture of, and I'm going to have to draw it. What are these? If you said the myelin sheath, you're correct. Good job. And what is specifically this right here? If you said node of Ranvier, you're correct. Let's write that down here. It's the node of... This is going to take forever. You can feel free to skip ahead until I have written this all out. Node of... It looks like Ranvier. It's pronounced Ranvier. Presumably it's French. Node of Ranvier. Okay, so that's what that is right there. The node of Ranvier. All right, we're done with the neuron. And again, I've said it already. I will say it again. The neuron can be in either the CNS or the PNS. Sometimes they go between both of them. Okay, but these next cells are in one or the other. Up here, we've got satellite cells and Schwann cells. The Schwann cells are like the myelin sheath, but in the... Oh, that's why I didn't have it on that neuron. See, I knew what I was doing. I just then didn't. So the neuron without those specific myelin sheath is going to be in either the CNS or the PNS. But depending on the type of myelin sheath it has is telling us whether it's in the CNS or the PNS. So we're looking at Schwann cells down here. These are the Schwann cells. The Schwann cells are essentially the myelin sheath in the peripheral nervous system. What is different about the myelin sheath in the peripheral nervous system from the central nervous system is that in the peripheral nervous system, each one of these Schwann cells has its own nucleus. In the central nervous system, there's one other type of cell that controls multiple myelin sheaths. So Schwann cells, each one is a different part of the myelin sheath. It's only one. There's just one Schwann cell to one nucleus, right? Okay, the satellite cells, these right here, these are around, these are kind of like astrocytes in the peripheral nervous system. They maintain that blood-brain barrier that we've been talking about. They also help cushion neurons in the peripheral nervous system. So we've got satellite cells, Schwann cells. Those are PNS. And now the central nervous system cells, we have this right here. These are ependymal cells. Those produce cerebral spinal fluid, of course. We've got this here. This is the ogliodendrocyte. And notice that it's a little light on my screen. I'm not 100% sure that I can see this. I've got two screens, though. It looks a lot better on one screen than the other, so hopefully you can see this. But this ogliodendrocyte is connected to 4 different myelin sheaths. So the ogliodendrocyte is the cell that is producing the myelin sheath in the central nervous system. All right, and that isn't a one-to-one. One ogliodendrocyte can support multiple myelin nodes, myelin sheaths. Okay, next we've got an astrocyte, the most cool cell, I think. Next to a neuron. Neurons are pretty cool. Astrocytes are just kind of like the unsung heroes of the brain. Anyway, the astrocyte is here, and then we've got this microglia, which is right next door. The astrocyte, of course, helps maintain the blood-brain barrier. The microglia helps clean out waste products. Okay. How does a neuron fire? A neuron fires. There's a couple answers in here that I want you to know. So first is it's all or nothing. It's not like it can fire stronger or weaker. It's just a firing. It's all or nothing. The way that it fires stronger is that it just fires more often. So a neuron will go like bing, 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 bing. And that means that it's very excited. If it's not very excited, it'll just go bing. But the bing, each firing, is the same amount of electrical charge needed, the same type of neurotransmitter is released. And so it's an all or nothing firing. Secondly, what I want you to know is it is an electrochemical process, right? So the electrical part is mostly happening within the neuron itself. It's all electrical charge coming from the dendrites, essentially. I mean, the dendrites are receiving the... I've just said this word like a million times. The neurotransmitters from other cells, and that creates an electrical charge. That electrical charge flows down the axon to the terminal buttons and then releases the neurotransmitters again to another cell. That's the electrical part. I mean, you can't have a firing without the electrical part. You can't really have a firing without the chemical part either, because the way that these neurons are talking to each other is also the neurotransmitters, releasing the neurotransmitters from the terminal buttons. So it's an electrochemical process, and they're not separable from one another. How do EPSPs and IPSPs work? EPSP is excitatory postsynaptic potential. IPSP is inhibitory postsynaptic potential. And that just means that it is changing the basically it's changing the threshold for the cell to fire in a way. The threshold is the same, and the EPSP is just bringing the base level of charge of the neuron up closer to the threshold. And the IPSP is bringing the base level of charge in the cell below the threshold. So they're making it easier or harder for the neuron to fire. If you have a bunch of EPSPs, just the tiniest change in whatever the cell is looking for is going to make it fire. Let's think about this in terms of hearing. If you are listening for something, you're directing a lot of attention to your auditory system. You're really listening for a sound. That's putting some EPSP into the neurons in your brain in the auditory cortex. They're going to fire more readily. If you know about the region of frequency that you're listening to, that can even help even more. So you're paying attention to a sound. You know that it's going to be a very high-pitched sound. So you're putting more EPSP into neurons that are responding to high-pitched sounds. So you're kind of like making it easier for you to hear what it is that you're looking for. Well, because of that, occasionally we can get false alarms because a little bit of information is going to make the cell fire. And so sometimes you end up thinking that you heard your name in a crowd when you didn't. Right? The typical cocktail party effect kind of thing. Okay, that's EPSPs and IPSPs. That's also the review. All right. Well, good luck. Of course, ask any questions that you still have after this through email to me and the TAs, and we will do our best to answer quickly. All right. Hopefully this was helpful. Good luck on the exam, and I will talk to you next week.